Good morning. Myself, Professor N.S. Gogre, Department of Soil Science, Kikewa College of Agriculture, Nasik. Today, we discuss on the topic nutrient use efficiency. What do you mean by nutrient use efficiency and how do you manage nutrient use efficiency? Before cultivating any crop in any season, we have to restore soil fertility. The ways of restoring soil fertility are addition of organic manure, artificial fertilizers, biofertilizers, and we manage the nutrient dose, that is recommended dose for all nutrients of all crops. If we take a certain crop, there is a recommended dose. We have to supply these recommended dose from soil level plant nutrients and adding additionally by soil testing of nutrients by the ways or means of organic matter, organic manures, artificial fertilizers, and biofertilizer. And what is nutrient use efficiency? The nutrients available in the soil after application of nutrients are utilized in crops. That is called as a nutrient use efficiency. Then we know about definition of nutrient use efficiency. Let's study. Nutrient use efficiency is defined as amount of dry matter produced per unit of nutrient applied or absorbed. Whatever nutrient we add, or what percentage of nutrients are absorbed is called as nutrient use efficiency. It is denoted by physiological efficiency into apparent recovery efficiency. Then there is various classification of nutrient efficiency. Agronomic efficiency can be defined as it is economic production obtained per unit of nutrient applied and calculated by the equation agronomic efficiency is equal to grain yield of fertilizer applied in crop in kilogram minus grain yield of unfertilized crop in kilograms divided by quantity of fertilizer applied in kilograms. It is agronomic efficiency. Then next, physiological efficiency. It can be defined as biological production obtained per unit of nutrient applied. It is calculated by the following equation. It is denoted by physiological efficiency equal to total dry matter yield of fertilizer crop in kilogram minus total dry matter yield of unfertilized crop in kilograms divided by nutrient uptake by fertilizer crop in kilograms minus nutrient uptake by unfertilizer crop in kilograms is physiological efficiency. Then apparent recovery efficiency can be noted by quantity of nutrient absorbed per unit of nutrient applied. Can be calculated by following equation. Apparent recovery efficiency is equal to nutrient uptake by fertilizer crop minus nutrient uptake by unfertilizer crop divided by quantity of fertilizer applied. It is all about apparent recovery efficiency. Then for efficient absorption or increasing efficiency of nutrients, we have to add fertilizer based on various timings. That is called as a time of fertilizer application. First, we have to add prior to sowing. Prior to sowing, some water insoluble fertilizers such as rock phosphate and basic fertilizer are applied 2 to 4 weeks before sowing in order to increase efficiency of fixed phosphorus so that it will come into soluble form and can be easily uptake by plant. Then at sowing, application of fertilizer at the time of sowing is called as a basal application. It is a part of recommended nitrogen fertilizer and entire quantity of phosphatic and potassium fertilizer applied as a basal dose. That is at the time of sowing. But we have to add nitrogen fertilizer in split doses to avoid volatilization losses of nitrogen, leaching losses of nitrogen. It can be applied in split doses for doses at the time of sowing. And next, after the sowing of the crop, application of fertilizers after the crop established is called as a top dressing. Depending on the stage of crop, a portion of nitrogen is applied as a top dressing. 1/3 dose of nitrogen is applied at the time of top dressing. 1/3 dose is applied at the time of sowing. Then number 4, that is known as split application. When fertilizer is applied at 2 or more different times, the system is called as a split application. It is applicable for nitrogen and it can be applied in 3 times, 3 split applications. Okay, next. Methods of fertilizer application. For efficient uptake by plant, there are various methods of fertilizer application. Number A, that is soil application. In soil application, there are various methods, of which number 1 is the broadcasting. Application of fertilizer uniformly on the soil surface is known as a broadcasting of fertilizer. Can be done before sowing of the crop or when the crop is in standing. That is called as a top dressing. It is widely practiced in India due to its easiest in application. Okay. Then band placement. Application of fertilizer near to the roots of soil is called as a band placement. And it is one of the most efficient method for application to soil. When fertilizer material reacts with soil constituents, leading to fixation, and where volatility losses are high, band placement is followed for efficient recovery of nutrients. Okay. The next method, that is point placement. Placement of fertilizer near the plant, either in a hole or in a ditch or depression, followed by closing or covering with soil, is known as a point placement of fertilizer. The number 4th method is fertigation. Application of fertilizer through irrigation water is known as a fertigation. It saves the application cost and allows utilization of relatively inexpensive water soluble fertilizer. It is generally followed by drip irrigation. Then application to the plant. Root dipping, foliar application, application of fertilizer to the foliage of the crops or leaves in a form of spray solution is known as a foliar spray of fertilizer. If there is a wide deficiency, it can be corrected by foliar application of nutrients. Then integrated nutrient management. Integrated nutrient management is a whole soil approach in which all 3 possible sources of nutrient application or supply is utilized. It can be used as organic sources, organic manures, inorganic sources such as artificial fertilizers, and biological components such as biofertilizer are used in integrated manner. Advantage. Enhances the availability of applied as well as native soil nutrients. They need synchronized nutrient demand of the crop with nutrient supply from native and applied sources. Provide balanced nutrition to the crops. Minimizes the antagonistic effect. What is antagonistic effect? It is when deficiency of one nutrient causes toxicity of another nutrient. Is the nutrient imbalance is called as a antagonistic effect. Then improves and sustain the physical, chemical, and biological function of soil. Then fertilizer grade. Fertilizer grade is an expression referring to the legal guarantee of the available plant nutrients expressed as a percentage by weight in fertilizer. For example, 12, 30, 16 is a fertilizer grade of NPK. That means it indicates the presence of 12% nitrogen, 32% phosphorus, and 16% potash. In fertilizer mixtures, various fertilizer mixture, that is open formula fertilizer mixture and closed formula fertilizer mixture, can be used. Open formula fertilizer mixture, that fertilizers are uh, prepared by dealers, which where it is mentioned that it indicates particular mixtures are mixed with this. It is called as open formula. And closed formula fertilization, it is prepared by farmer at his farm level, whether which contents of fertilizer mixture, that is NPK, are not disclosed, is called as a closed formula fertilizer mixture. Then urea can be mixed with all types of fertilizer. Other fertilizer can be mixed just before use, that is closed formula fertilization. But the efficiency of the following fertilizers will be lowered with the mixed fertilizer. Then fertilizer use efficiency. It may be defined as yield per unit of fertilizer input, or it may be fresh weight of or product yield per unit nutrient content. Then there are different nutrient efficiency of different nutrients, depend on their mobility or immobility. The nitrogen, which is very mobile, its efficiency 30 to 50%. It is cause of low efficiency is immobilization of nutrients, conversion of inorganic form again into organic form, complex organic form, then volatilization losses, denitrification, and leaching losses. Then phosphorus efficiency is up to 15 to 20% because phosphorus has a tendency to get fixed in soil. Then its efficiency is decreased. In potassium, it has high efficiency, that is 70 to 80%. It has a limitation of fixation in clay only. Then sulfur, 8 to 10% efficiency. It is due to immobilization, leaching with water, and micronutrients. Fe, zinc, Fe, copper, boron, molybdenum, and manganese. It has efficiency of 1 to 2% because it gets fixed in soil. Then factors affecting fertilizer use efficiency or nutrient use efficiency. That is leaching, NO3. Leaching means fertilizers or nutrients get leached below the root zone. Then number 2, that is gaseous losses of NH3, NO2, and N2. In immobilization, it is by chemical precipitation, absorption on exchange complex, and microbial cell. It is conversion of inorganic form into organic form, that is immobilization. Then various chemical reaction between various components in fertilizer during mixing before application to the soil. Then physical properties of soil affects fertilizer use efficiency, and also chemical properties of soils. Adverse in chemical properties of soil, adverse in pH, EC affect the fertilizer use efficiency. Then fertilizer characteristics. Nature of fertilizer, that whether it is acidic fertilizer or basic fertilizer, affects fertilizer use efficiency in case of a soil which differ from soil to soil. Then methods for increasing fertilizer use efficiency. For increasing fertilizer use efficiency, two methods are followed. That is known as agronomic method and chemical method. Agronomic method. In this, application of fertilizer at right time with required quantity can be useful for increasing fertilizer use efficiency. The phosphates are more efficient when the entire dose is applied at basal dressing. But potash entire quantity as a basal dose is applied, and nitrogen is applied in split doses, that is 2 to 3 doses. Example, sugarcane. Potassium application normally done along with nitrogen application. On better utilization of nitrogen in the presence of potassium. Therefore, K is applied at 45 to 90 days application, 90 days after sowing. Then chemical methods. Synthetic or chemical fertilizer should be good quality and right source for the crops. Examples, oilseeds, tobacco. Tobacco plant avoid using of fertilizers containing chlorine for preventing from scorching effect. In tea plantation, use of ammonium sulfate is best. In paddy, ammonium and urea fertilizer are more efficient for paddy under different India conditions than nitrate fertilizers. Okay. Is there any question? You can write it on paper or ask me after the COVID period is over. This is all from my side about nutrient use efficiency and management of its nutrient use efficiency. Lip analysis, standards and index tissues of different crops for interpretation of lip analytical values. Every time during cultivation of any crop, we have to restore soil fertility. Generally, we use this soil available nutrients, and if recommended dose is not sufficient, soil available nutrients are not sufficient to meet that recommended dose, then we add the rest of the nutrient dose by artificial application of organic manures, artificial fertilizers, and biofertilizers. So that total recommended dose of fertilizer, which is optimum for undertaking cultivation of any crop, is recommended, and we have to fulfill that dose. But out of the recommended dose from soil available nutrients and by additional application of nutrients by these sources, how much amount of nutrient is taken can be efficiently analyzed by analyzing it through various growth stages. The lip is the source which can mask the availability of nutrients, uptake of nutrients from soil. So we generally follow plant analysis. In plant analysis, the main importance is on leaf analysis, or it is also called as a proximate analysis. There is a precise method of monitoring plant nutrient levels, that is particularly from stem leaf analysis or tissue analysis is the most important factor which determines the uptake of nutrients. When soil reveals the levels of essential nutrients, leaf analysis shows the growers or cultivators exactly what the plant has successfully absorbed or uptake. that amount can be easily determined by leap analysis. So it has a significant importance in determination of plant analysis or leap analysis. To predict, leap analysis is also done to determine whether there is a deficiency of nutrients, or we can correct the deficiency of nutrients by foliar application, because the nutrients deficient can be easily utilized by application of foliar spray. Okay. Then, what is the importance of undertaking leaf analysis, in particularly plant analysis? It is a chemical analysis of plant foliage, of which important tool for establishing and maintaining proper fertilizer program. To undertake proper fertilizer means we have to restore fertilizer or a rest of nutrient which is not available in the soil. We can meet the deficient or deficit in the form of fertilizers or organic manures for efficient or quality or production, efficient production. Leaf analysis can be also used to confirm or diagnose problem associated with nutrient application. Means if there is a deficiency of a nutrient, it can be corrected by leaf analysis. By leaf analysis, we are able to draw the conclusion whether there is a problem associated with deficiency of nutrients or toxicity of nutrients. Then shortages or excess, and more importantly, to prevent the development of nutrient disorders in crop. There are various deficiency disorders of nutrient, which occurs due to deficiency of nutrient in the soil, so that it shows its symptoms on plant. Particularly, this includes in tomato, there is a particular disorder that is called as a blossom rate in tomato. It is due to deficiency of calcium. It is a disorder by deficiency of nutrient that is known as a calcium. If there is a deficiency or a disorder, then we can prevent it by application of calcium in advance and minimize that deficiency by undertaking foliar spray at initial stages, because later it cannot be corrected. If there is development of flower, then there is no use of any such correction of deficiency. In another disorder that occurs in cauliflower, it is called as a whiptail disorder of cauliflower. It is due to deficiency of molybdenum. If there is a deficiency of molybdenum, we can easily detect deficiency of molybdenum by undertaking leaf analysis, and then we will be able to correct that deficiency of molybdenum with the help of foliar spray of molybdenum at 2 mg per liter. That is 0.2 ppm to recover the deficiency of molybdenum or to avoid disorder like whiptail in cauliflower. In another deficient disorder of boron, that is in the crop called as a pomegranate. The cracking in pomegranate is due to deficiency disorder due to deficiency of boron. Then good luck. Hello.
हॅलो हा सर.
काय प्रॉब्लेम काय? तुम्ही चॅटिंग का करताय?
नाही सर, टॉपिक विचारला होता मला, मग मी सांगत.
कोणी विचारलं? बरं ठीक आहे. ते काय होतं, डिस्टर्ब होतं. अशा चॅट न करू, कारण चॅट चेक करायला लागतं ना.
हो सर, सॉरी.
ठीक आहे, ओके. Okay. Then next disorder due to deficiency of nutrient. Sorry. A deficiency disorder of nutrient, that is Khera disease in rice. It is due to deficiency of zinc nutrient. So we can well predict or analyze the deficiency of zinc, so that we are able to prevent this deficiency disorder well in advance. Okay. Then this will also reveal certain fertilizers are being used and are not necessary and results in most economical fertilizer program. We can easily diagnose or easily reveal that certain fertilizers don't have any impact on efficient utilization of economical fertilizer and efficiency. So we were able to avoid that and minimize the cultivation loss, cultivation cost. Analysis must be properly taken. For this, index tissue is most important to arrive at a proper explanation or to do the paired comparisons or to follow the statistical tests, which are frequently helpful for undertaking that leap analysis. We have to produce the data from various observations and by recording this observation, we are able to conclude whether the test is acceptable or not. Okay. Foliar analysis made over a period of years increases or indicates approaching deficiency of nutrient element before plant shows any visible symptom. That is explained previously. Before occurring deficiency of any nutrients or to prevent deficiency of nutrients from foliar analysis of leaf from leaf analysis, we have to predict or undertake the foliar spray. So we were able to avoid the deficiency or whether plant shows these deficiency symptoms or visible symptoms. Then it is this toward the foliar spray to undertake foliar spray. It can be possible through proper corrective fertilizer application to prevent the deficiency from ever occurring in the crop. We have to predict proper spray of fertilizer to avoid the deficiency in crop. It is possible to learn when an element may be increasing in a crop
toward a level that will be toxic, and we limit the quality of that plant or quality of that produce to avoid undesirable effect only by leaf analysis. When this condition is known, states are able to taken to alter the fertilizer program, where we can change the fertilizer program or undertake some cultural practices that influences the uptake of element from the soil. We adopt certain practices or follow certain practices to increase the efficiency of soil available nutrients. Like if there is a deficiency of phosphorus, if there occurs a deficiency of phosphorus, then we have to increase the efficiency of phosphorus, which is already present in soil. Phosphorus has a tendency to get fixed in the soil. So we can increase the efficiency of this fixed phosphorus by undertaking biofertilizer program. That is, phosphate solubilizing bacteria can be applied to the soil to increase the efficiency of that particular fixed phosphorus. There is no need to apply phosphatic fertilizer. We can easily reduce the production cost and increase the availability of fixed phosphorus and increase the phosphorus use efficiency. Then it is one of the most important fact that what plant analysis shows. Like soil analysis mark the availability of nutrients that are in a soil, but plant analysis marks the uptake of nutrient status of plants at the time of sampling. Simply, it denotes the uptake of nutrient from soil through various processes, that is known as mass flow and diffusion. And that particular nutrient concentration is denoted in plant tissue. Or it shows whether soil nutrient supplies are adequate or not. Or it will detect any unseen deficiencies, as we discussed previously, by confirming some visual symptoms of the deficiencies that can be corrected. Then we can also arrive at conclusion by plant nutrient status, by plant analysis, whether there is any toxicity of nutrients detected using a diagnostic tool. Plant tissue analysis from M plant will able to derive conclusion of deficiency symptoms. Then we can follow corrective fertilizer application program during that particular season. So, not all abnormal life processes are due to deficiency. There are certain symptoms which are abnormal and which are not due to deficiency of nutrients. We have to briefly study that. So that, but deficiency symptoms are specific, and we can determine or draw a conclusion. Some deficiency symptoms may be due to much of certain elements. Symptoms of one deficiency may look like others. but there is a deficiency for a particular nutrient. They look like similar, but it can be easily diagnosed. We can take an example. The deficiency of an iron and nitrogen is similar, which produces pale yellow color or chlorosis. But deficiency of nitrogen or macronutrients or major nutrients which occurs on older leaves, and deficiency of micronutrient which occurs on younger leaves. So we can easily detect which nutrient has deficiency. Means if it is occur on older leaves, then it is deficiency of nitrogen. If it is on younger leaves, then there will be deficiency of ferrous or iron. If plant comes from the field, it is always affected with weed, insect, and disease organism. Then we can easily detect the moisture stress and mechanical injury that are not deficiency symptoms itself. Then interpretation. The sufficiency ranges are given for plantation of crops at stages, that is the crop growth stages. That research has been found appropriate for sampling. Then there is a particular procedure for undertaking the sampling. Then analysis data can be used as a guide for attaining improved quality and yield. We know which element is for better quality of fruit crops, whether it is horticultural crops. Then by correcting these deficiency, we are able to draw satisfactory yield. For long-term orchard crops, plant tissue nutrient levels can be logged over time and used as a basic diagnostic data or diagnostic tool to assist in a development fertilizer program. Take an example of banana. The tissue analysis or leaf analysis is the most important for banana crops because of its broader leaves and efficiently uptake of nitrogen, because these crops heavily require the nitrogen from soil, or it facilitates on or increasing uptake of nitrogen over the time. We have to lodge or would have to follow the leaf analysis at particular growth stage, that is pre-sowing, sowing, top dressing of fertilizer. These are 3 stages before flowering and before fruit set. These are important growth stages. At these stages, leaf analysis help to increase quality of the fruits and production also. If a tissue level of a nutrient is below lower end of sufficiency range, then it can be considered as deficient. We can meet this deficiency by addition or correction of whether the source is artificial fertilizer immediately or organic fertilizer or biofertilizer over the period. We have to predict or target yield in particular commercial fruit crops like banana. We able to drive targeted yield concern, that is known as soil testing and crop response, by doing the analysis, soil analysis, and alternate taking plant analysis at various growth stages. We predict or target the yield, and accordingly, fertilizer program can be developed to achieve the desired production or targeted yield. Then at the level approaches, our limit, that is addition of nutrient, should be withheld. Means we have to stop addition of the nutrients at a particular peak because it is important to be near midpoint for most important nutrients because the imbalances in the ratios of nutrients can affect crop. Environment plays a major role in nutrient uptake and crop development. The sufficiency ranges given here should be considered as a general guides. In addition to variation, due to various environmental effects, different crop cultivars have different critical levels of nutrients. Then we have to diagnose these critical levels of nutrients by diagnostic diagnostic recommendation and integration system. It is all about leap analysis. If there is any question, you can simply turn on your mic and ask the question. If there is any question to this topic, you can turn on your mic and answer the question if there is any question. Okay. But like you can turn on in your mic.
Hello.
Have you got the question paper?
No, sir.
So I have searched the question paper. आम्ही सुद्धा क्वेश्चन पेपर बघितली मला काही सापडले नाही कारण कॉलेजमध्येच असेल आम्ही गेलो होतो त्यासाठी कॉलेजमध्ये सर प्रिन्सिपल सर पण नव्हते आणि सगळ्या लोक होते त्यामुळे मला काही ते मिळाले नाही आता 2-3 दिवस बाकी आहेत त्यानंतर काय होईल माहित नाही मला पुढचं सांगता येणार नाही पण तरी पण बघा ना कोणाकडे तरी मिळेल का
हो सर विचारतो परत मी भेटलीत
बघा कारण कसं आहे आता तुम्ही एकच क्वेश्चन एकच झालंय एकच वर्ष झालंय एकच क्वेश्चन पेपर आहे आणि हा सब्जेक्ट म्हणजे पहिला हा थेरोटिकल प्रॅक्टिकल होता त्यानंतर थेरोटिकल झालाय तर ठीक आहे नेक्स्ट लेक्चरला जर नाहीच मिळाली तर मी तुम्हाला काही क्वेश्चन देईल ते मी ERP थ्रू तुम्हाला मेल करून टाकेल तुम्ही ते पर्टिक्युलर टॉपिक्स सर्च करा आणि आतापर्यंत जेवढं झालं मी ते तुम्हाला सगळं पाठवलंय, मेलवर पाठवलंय बरं. ते राहिलं ते पाठवतो मी. ओके.
हो सर.
ओके
हो.
ठीक आहे. तर आपण इथे थांबूया, चालेल.